Well, hello everybody, and a very warm welcome to you all on behalf of the NC3Rs, the 3Rs Prize Panel, and the prize sponsors, GSK, to our celebration of this year's 3Rs Prize. My name is Julia Buckingham, and I'm the chair of the NC3Rs, and I'm here today with Natalie Duggett, who's played a very important role within NC3Rs in running the prize this year, and she and I are going to host this jointly. Now, I'm really thrilled that we've got the opportunity to come together to celebrate the wealth of 3Rs research, which is taking place right across the globe, and the really brilliant science that's being delivered, and of course, this very important prize. A huge thank you to GSK for their very generous sponsorship of this. This afternoon, we're going to hear from our sponsors, GSK. We have a short presentation. Then we're going to hear from our 3Rs prize winner, Dr. Francesco Nevelli from Merck, and our highly commended recipient, Dr. Lorna Ewart from Emulate. Francesco is with us here online, and he will be available to answer questions after his presentation. So if you have any questions for him, please ask them at any time during the presentation using the Q&A function at the bottom of the screen. Now, unfortunately, Lorna's traveling, so she couldn't join us in real time today. But her presentation is a recording, but she has indicated that she will be happy to answer any questions that you've got, and we'll let you know a little bit later on how you can go about asking them. Now, as I'm sure you all know, the 3Rs Prize recognizes and rewards outstanding and original scientific research that has or could have a major practice-changing impact on the replacement, the reduction, or the refinement of the use of animals in research. The prize is awarded annually for a piece of primary research which has been published in a peer-reviewed journal in the last 3 years. It's open to any researcher anywhere in the world, so it's a truly global prize, and they can be in industry or in academia. And the prize can be awarded to the principal investigator, the research team leader, or another nominated author. Now, this year is the 20th birthday of the NC3Rs. It's 20 years since we were established, and we've been celebrating this landmark by showcasing some of the key achievements that the organization has made in furthering the 3Rs since it was formed in 2004. And you can find details of that on our website. Now, insofar as this prize is concerned, this is the 19th 3Rs prize that the NC3Rs have awarded since we launched it, with very generous support from GSK right from the beginning. So we really are hugely grateful to them. And thank you to everyone who submitted applications this year. We had 31 applications from 13 different countries. Now, as I'm sure many of you will know, the judgment of the prize is based on 4 main criteria. There's the actual or potential 3Rs impact of the research reported, the quality of the published research, and that includes the importance of the research question and the transparency and the robustness of the study design. How well the 3Rs implications have been disseminated to date, and the strength of the plans for the prize grant, including potential impacts for the research on the 3Rs and the impact on the development of the individual. Now, the people who make the decision, the prize panel, was made up with individuals from the UK, Europe, and North America, and I'd like to extend my thanks to all of them for their help in determining the winner. It's a lot of work, and their commitment and their professionalism is absolutely fantastic. It's also a very tough job. We had a number of very impressive applications and some really great discussions before we came to our unanimous conclusion on the outcome. So huge congratulations to Francesco and to Lorna for their really brilliant work. Now, I'm now going to hand us over to Dr. Gina Wilkinson from GSK, and we're going to hear a few words from her. Gina is the Chief Veterinary Officer at GSK, and she's just going to tell us a little bit about their perspective on the prize. So, Natalie, I think you're going to get the recording going.
I am thrilled to represent GSK in the recognition of the NC3Rs International Prize, celebrating excellence in global science that contribute to the replacement, reduction, and refinement of animal use in research. This marks GSK's 19th year as a funding partner for this outstanding award. At GSK, we are uniting science, technology, and talent to get ahead of disease together. Our ambition over the next 10 years is to positively impact the health of more than 2.5 billion people. Our strategy is centered around preventing and treating disease, focusing on the science, and operating responsibly. In pursuit of our strategy, we have leveraged many new technological advances or non-animal methods to advance our research. These methods, however, do not always provide the exact insight needed to determine how a potential vaccine or medicine affects disease mechanisms or biological systems. Therefore, animal studies are still needed. Our long-standing commitment to the NC3R's prize aligns with our strategic direction and our own 3R's ambitions, as they are an integral part of discovering and developing transformative medicines and vaccines for patients. Supporting and recognizing innovative and groundbreaking research through the NC3R's International Prize is what we consider doing the right thing. We are truly proud to be a part of an award that will help to showcase high-quality science and inspire future advances towards replacement, reduction, and refinement of animals in research. Personally, I would like to congratulate and thank all the entrants, as you are all winners in the pursuit of research excellence.
So we come to the highlight now, hearing from our prize winner, Dr. Francesco Nevelli from Merck. Francesco and his team have published a paper which is entitled Biological Assay to Determine the Gonadotropin Potency from In Vitro to In Vivo Sustainable Model, was published in the International Journal of Molecular Sciences in April 2023. Now, this winning paper describes an in vitro assay to test follicle-stimulating hormone potency. The method is approved by regulators for use in potency testing, and it has the potential to replace tests in tens of thousands of rats annually, so a quite extraordinary replacement potential. The panel commended the development of the in vitro assay and the work that Francesco and the group at Merck have done in pursuing regulatory approval. The current and the future 3R's impacts were considered to be significant, given that over 70,000 IVF cycles were reported by the Human Fertilization and Embryology Authority in the UK alone, and the group are actively working towards worldwide acceptance. So, Francesca, we very, very much look forward to hearing your presentation, and I'm going to hand over now to you.
Good afternoon to everybody. First of all, thanks a lot for this amazing prize. We are really honored, myself, all the authors and the team, to receive such a prize. This paper describes a part of a bigger project that we started 10 years ago, and that is aimed to improve the sustainability and the quality of the control strategy for our fertility product. And this prize, it's a lot of fuel and a further motivation to go through in this project. So the focus of the paper, it's a comparability study between an in vivo and an in vitro assay to be used to quantify the biological activity of follicle-stimulating hormone. Biological activity or potency or efficacy, it's a mandatory quantification prior to the administration to the patient of each batch of each product. The potency is quantified through bioassay that are analytical methods in which a biological system is stimulated by the drug to produce a dose-related response. These methods are product-specific. Indeed, we have to develop a dedicated method for each product, and they should reflect the mechanism of action of the molecule. And this could be pretty tricky, especially in the case of the in vitro method. Follicle-stimulating hormone is broadly quantified with an in vivo bioassay that is described in the European Pharmacopoeia, and it assesses the potency of the hormone through the ovarian weight increase quantification after the stimulation of female rats with FSH. Some years ago, in our laboratories, we developed an in vitro bioassay that exploits a cell line that overexpresses the follicle-stimulating receptor to produce a cell-specific metabolic cascade. Of course, this in vitro method was developed in order to replace the in vivo method. The in vitro method was validated in our lab, and plenty of batches were tested, release and stability batches, in order to test what was the behavior of the method in the real life. But the most important step was the demonstration that in vitro and in vivo method could behave in the same way, that both methods can guarantee the same control level for our product. Indeed, we had to compare a simple method, like the in vitro one, with a very complex method, like the in vivo, that could mimic the whole life of the molecule, starting from the injection route through the metabolism, the delivery, and the interaction of the drug with the target. In other words, we have to demonstrate that the in vitro method has the same ability in detecting chemical and structural modification that could occur to the molecule in the same way as the in vivo one, because this chemical and structural modification could impact the metabolism of the molecule, so the half-life, and finally, the biological activity of the molecule. To do this, we applied several chemical and physical stress to 3 different batches of follicle-stimulating hormone in order to obtain different cellulation level, sample at a high level of free subunit, and sample with high oxidized variants. All these modifications could indeed impact the metabolism. These samples were chemically and physically characterized, and then were tested against the untreated sample in order to identify if both methods were able to discriminate this kind of modification. In particular, if the in vitro method would be able to reject sample with non-compliant level of this different modification. You can see on the left-hand side of the slide the results. For both high free subunit and oxidized variants, both methods demonstrated to be sensitive and able to discriminate the difference in the same way. Moving to cellulation level, we produce sample at 50%, 75%, and 100% of cellulation. And we compare the result of those samples with the untreated one. For both methods, we found a strong correlation between cellulation level and potency. In addition, as you can see from the graph on the bottom corner of the slide, the 2 methods showed an opposite effect of cellulation on the potency. This was expected and also described in literature. But for us, for our goal, this was not important, because we wanted to demonstrate that both methods are able to reject sample with non-compliant cellulation, and this study demonstrates that. To conclude, the 2 methods demonstrated to be able to discriminate and to detect the difference at the level of the quality attribute of the molecule. So the in vitro method can be considered suitable to replace the in vivo one. These results were the green light to start the submission rollout for the change of the replacement from the in vivo to in vitro method. Currently, we already obtained the approval of about 70 countries all over the world, in particular from Europe and UK. The impact of this project could be really dramatic in terms of animal replacement. Our estimation is to reduce of about 20,000 animals per year, starting from 2026. And to completely replace the use of animal in this kind of activity, starting from 2032. This would mean about 40,000 animals per year. In parallel, the replacement of the animal use would also lower the energy consumption in our facility over the 20-25%, comparing the animal facility with the in vitro one. This project is fully in line with the One Merck animal welfare strategy. Indeed, in Merck, we are committed to innovate and develop alternatives to supersede the use of animal in all the different divisions. And the strategy is fully supported by our CEO, Belen Garrido. How do we want to exploit the prize, the grant prize that we receive from NC3R? Funding educational program for physician and patient based on the ethical implication of the drug development and the analytical development in order to guarantee the quality, the safety, and the efficacy of the product. And to improve the knowledge in order to perform informed decision for patient care, still based on the quality of the product. Or to explore opportunities for funding student secondment in order to develop new, innovative, and animal-free model for quality control for the whole life of the drug, and to implement artificial intelligence in the analytical development. My last slide is an acknowledgement to all the paper authors. We are a cross-functional team coming from different departments of Merck, from different sites, with different expertise, and for me it was really a pleasure to work with all of them. I learned really a lot in this experience. And a special thanks goes to Dr. Dadone. We have been working together for more than 10 years on this project. We shared everything during this year from a professional point of view. We grew up together, we learned through each other. We had to face with a lot of challenges, but we never gave up. So thanks. And thanks to all of you for your attention. And I'm pleased to answer to all your questions.
Brilliant. Thank you very much, Francesco. It's really interesting. We have a couple of questions. I just wondered if you could tell us a little bit more about kind of Merck's next steps towards getting that worldwide regulatory approval for the in vitro test here.
From the regulatory point of view, we are facing the submission rollout in a stepwise approach. Indeed, we started from follicle-stimulating hormone with the drug substance request for the switch, and then we will move on to the drug product, and this is based on the kind of registration that each country is request. And then we will also move to the other fertility product that we have in our portfolio, and the timeline for the replacement are the one that I showed you in my presentation.
Thank you. Thinking about the other products that Merck produce that contain FSH, we've had some questions in about kind of the wider potential for this assay, and about, FSH products we know are widely used in IVF for patients in the clinic, but also in other industries like agriculture and in the dairy industry, and whether there's a potential for those products to also, have this same kind of quality control process done in an in vitro way rather than an in vivo test as was previously done.
The plan is to move to the in vitro test for all the FSH-based product we have in the market.
Huge potential across, animals and human use for FSH products. And thinking about kind of the medical community, I thought it was really interesting the plans that you have for the 3Rs Prize and kind of informing that medical community about what else is out there that has been approved in different ways. Have you encountered much resistance from that field for products that have been approved without the use of animals at all?
Of course, I'm a lab researcher, so I don't have this particular kind of feeling. We are at the beginning of the journey. So I don't really have an answer for this question. Okay. I'm sorry, but it's a bit out of my knowledge.
No, that's fine. And thinking about kind of the wider strategy at Merck in terms of looking to replace the animal work that you guys do. Obviously, this is having a fantastic impact there, replacing 20,000 animals per year, going up to 40,000. Is the kind of general strategy across Merck to aim for more of these replacement approaches being developed and pushed forward?
It is a general approach. Yes, we have this one Merck animal welfare strategy that is aimed to replace as much as possible the use of animal. Nevertheless, this is a particular field in which the replacement is easier than other field. We are looking for a precise response to be analyzed, so it's easier, let me say, than in other study that have a more holistic approach on the response that should be kept under control. So this was the first project, the most advanced one, I think, in Merck now, but we have some advantages in that.
That's great. You're leading the strategy for the rest of Merck, and that's really exciting to see. Thank you, Francesco. I mean, Francesco will be sticking around till the end of today's prize event. So if you do have any more questions, please do feel free to send them through to us, and I'm sure you'll be happy to answer one or two more if they come through at the end, Francesco.
Thanks a lot.
Absolutely
available.
Brilliant. Okay. So without further ado, I'm delighted to introduce our highly commended winner this year. So the highly commended award this year has been given to Dr. Lorna Ewart from Emulate. The paper, Performance Assessment and Economic Analysis of a Human Liver Chip for Predictive Toxicology, was published in Nature Communications Medicine in December 2022. The paper describes the development of liver chips. These organ-on-a-chip devices were able to determine liver toxicity with a high degree of sensitivity, minimizing the need for animal models. The panel recognized this approach as highly innovative, demonstrating confidence in the utility of organ-on-a-chip technologies in the short term to help minimize the use of animals in drug testing, but also in the longer term to replace in vivo testing with complex in vitro approaches. I'll now share a short recording from Lorna.
Thank you, Natalie, and particular thanks also to the NC3Rs and GSK for the highly commended nomination for our work supporting the prediction of small molecule drug-induced liver injury using a human liver chip. So liver toxicity is a leading cause of drug attrition in the pharmaceutical industry. And here in this slide, we're showing a couple of examples of where we spot this drug attrition. On the left-hand side, you'll see it's a leading cause of clinical trial failure. But in actual fact, it's a very big problem when drugs go onto the post-market place. Here we can track different drugs over the last few years that have actually caused severe market failures and led not only to patient fatalities, but also in some cases, the need for liver transplantation. So this was really our motivation for designing a model that would allow us to predict small molecule drug-induced liver injury to ensure we have greater patient safety, both during clinical trial and beyond. Now, some people have told us that actually drug-induced liver injury, or DILI, is actually a solved problem. Well, at Emulate, we beg to differ on that argument, and this slide really shows why. Over the last 2 years, we've been tracking very high-profile examples where either clinical trials have been paused by the US FDA or other regulatory authorities, or indeed companies have discontinued a particular product during development. So not only is this costly for the pharma industry, it's also tragic for the patients who really need these particular drugs for their underlying disease conditions. So the work that we've been doing here at Emulate is to build a human liver chip. This model's been in development for many years. We first published in 2019 in Science Translational Medicine. But we've continued on our journey, and our latest publication at the end of 2022 took our human liver chip, which is shown here in this particular slide. What we were doing was we were placing human hepatocytes on our top channel of our chip. We have a chip that has 2 parallel microfluidic channels. We were placing those hepatocytes in a very traditional sandwich culture. This is commonplace for the polarization of hepatocytes. We have a membrane that separates our two microfluidic channels, which is designated as number 4 on the diagram here. And below in our smaller vascular channel, we're actually seeding 3 additional cell types: the human hepatic stellate cell, the Kupffer cell, and also the liver sinusoidal endothelial cell. Now, together, these 4 cells actually represent a very basic liver sinusoid, and we're trying to use our membrane in our chip to really represent that space of Dees. We can control the culture of these cells for up to 14 days in culture, and we show nice hepatic functionality. Now, the pharma industry really are interested in the development of microphysiological systems, or MPS, of which organ-on-a-chip technology is one example. And they actually have formed an affiliate organization known as the IQ MPS Systems Affiliate. Here, this is where a group of industry colleagues come together, and they actually provide guidance to organizations like Emulate and others around how one could build an effective liver-on-a-chip model for use in drug development. So we've actually followed one of their publication by Body et al. In 2020. And Body actually set out 3 main criteria or stages. First of all, they were asking developers to look at an albumin and urea production, as well as some gene expression. Next, they were asking us to look at biomarker stability, enzyme activity, and tissue histology. And finally, and more importantly, if we pass those 2 stages, could we then test compounds and show whether they indicated hepatotoxicity or not? So starting off then with the baseline characterization, we actually undertook this baseline characterization over 870 liver chips. So we believe it's one of the largest, if not the single largest organ chip study published to date. Starting off on the left-hand side, you can see some images under phase contrast, but also using immunofluorescence of the hepatocytes. So you can see that they're nicely cuboidal, they've got plump nuclei, you can see binucleated cells as well, very clear boundaries between the individual cells. And using the immunofluorescence, we can stain for the cytoskeleton, where you can start to see the formation of that bile canaliculi, as well as many mitochondria. As you know, the liver is an organ that requires a high degree of mitochondrial activity. It's a very large energy requiring organ. And on the right-hand side here, you can see the 3 human hepatocyte donors that we tested throughout our study, simply labelled here as donor 1, 2, and 3. And we follow these out for up to 7 days in culture on the chip, which is actually equivalent to 13 days in culture overall. And you can see that our levels of albumin production, we cite them in micrograms per day per million hepatocytes, sit nicely above a minimum range of about 20 micrograms per day per million hepatocytes, which would be the lower end of physiological relevance. The individual dots actually indicate the individual chip, so you can see there is a degree of chip-to-chip variability. Next, we were looking at key drug transporters and metabolizing enzymes that are important for the liver to demonstrate functionality for, and also play a role in various mechanisms of drug-induced liver injury. So the enzymes on drug transporters are labeled along the x-axis, and on the right axis, you can see the fold change relative to that same hepatocyte donor, but straight from the vial, so not on the chip under culture at all. And you can see at 2 different time points during culture, the light grey being day 3 and the dark being day 7, that we have good stable enzyme expression during the culture period, with the exception of 1 or 2 markers that are dipping slightly below that baseline level, indicating that further optimization may still be required to further enhance the performance of the chip. So we met those basic criteria set out by the IQMPS, and then we actually looked at how well the liver chip would do in terms of distinguishing between structural analogues of drugs. Now, if I take you back, first of all, to how the pharma scientists would be working here, at this stage in their project development, they'd likely have 2 or 3 candidates from the same chemical series, and they'd be looking at placing those in a rank order. So in other words, they want to progress the safest out of all of the 3 drugs available. So the way that we worked with the liver chip was we blinded our scientists to all of the drug identities, and here we're looking at particular pairing trovafloxacin and levofloxacin. These are both prescribed antibiotics. Trovafloxacin, though, is the toxic of the pairing. Levofloxacin is the lesser or non-toxic equivalent. And so by doing our experiment, we could look at albumin production, ALT, which is an enzyme released by the cell when it receives damage, and our proprietary morphology score, whereby a higher score would indicate a dead or dying cell. And what you can clearly see in the black circles, which represent increasing concentrations of trovafloxacin, that there's a concentration-dependent reduction in albumin, a concomitant increase in ALT, and also an increase in the morphology scoring. So this would indicate to our scientists that this was a hepatotoxic drug. On the open circles, what you can see is levofloxacin, and you can see no change in albumin, ALT, or morphology during the course of this particular experiment. Now, the IQMPS had actually set forth a number of different paired analogues for developers like ourselves to test. And here we've listed all of the pairings that we looked at with some of the key data points. And you will see that in each occasion, and again, I remind you, this was all done under blinded scientists and also verified by external toxicologists who were also blinded to drug identity. You can see liver chip correctly identified all of the toxic drugs on the left-hand side of the table, and from the middle towards the right-hand side of the table, you can see they also correctly identified the lesser toxic drugs. So we were really very pleased with this particular outcome. Now, in addition to the structural analogues, we actually tested a number of drugs, and this slide really summarises our quantitative assessment of we actually looked at 27 drugs overall, but 18 of them we looked across 2 particular hepatocyte donors, and we were asking the question, how sensitive were the readouts coming from liver chip and how specific they were. So maybe a couple of words before we actually dive into the data here. As a reminder, it is a requirement for pharma companies to test on 2 animal species prior to entering first in human. And all of the drugs that we tested had actually been in the clinic, so we knew the clinical outcome. So we knew if they were hepatotoxic or non-hepatotoxic. By definition, therefore, all of those drugs, all 18 in this example, have been through an animal model. Now, we don't have the animal model data, but what we do know is that of those 18 drugs, 15 of them were clinically hepatotoxic. Therefore, if the animal did spot that there was a potential signal with this molecule, it incorrectly associated the risk. As you'll recall earlier on, I mentioned some of the drugs had actually been associated with patient fatalities. So for that reason, we say that because animal models failed to identify 15 clinical hepatotoxicants, we'd actually give this model a sensitivity of 0% as our baseline. However, of the non-hepatotoxic drugs in this set, there were 3 of them, and we know that they correctly identified those. So we give it a sensitivity, a specificity of 100%. So when we then turned to the chip, we were absolutely astonished to see, and of course very happy to see, that of the 15 hepatotoxic drugs that went into human liver chip was able to identify 13 of them. It did miss 2. So that would give liver chip a sensitivity of 87%. And of course, we also were able to pick up the 3 non-hepatotoxic drugs, giving us a 100% specificity. So what does this really mean in reality? Well, an 87% sensitivity value actually tells us that the liver chip is really very good at detecting true clinical positive DILI compounds. So for every 7 out of every 8 hepatotoxic drug, the liver chip is going to be able to identify it. However, what's also very important to understand is that to get a sensitivity and specificity value, many researchers try and balance the two. So they'll look at the level to give them the highest sensitivity and the highest specificity, but often those two come against each other. So in this case, we actually set our threshold to read 100% specificity because we wanted to really understand the true sensitivity of our model. And by doing so, by setting our specificity to 100%, we can actually give the confidence to scientists that they won't be stopping a good drug during their clinical development program, e.g., they won't be having a false positive. Now, how does this help in terms of the pharmaceutical workflow and indeed the way that this could help in terms of a 3Rs agenda? So what we published was our typical workflow, which you can see in grey along the top, which is where a pharma company will use in vitro models and then enter into animal dose range studies and ultimately into the pivotal GLP tox study before going into the clinic. Now, of course, we know that this path is tried and tested, but sadly still does lead to around about a 90% failure rate for every drug that goes into phase 1 clinical trial. Only 1 out of every 10 are going to successfully make it into the marketplace. So what we're advocating is that if one were to use liver chip after maybe some of these higher throughput in vitro models, if liver chip was returning a drug that had a toxic signal, we clearly then offer a choice to scientists. They may choose, because there may still be a risk benefit or a cost benefit to doing so, to progress into animal studies. But indeed, they may also recognise that of their 2 or 3 candidates they're profiling, maybe only one of them is truly clean, which can then go through the animal studies and of course then go into the regulatory process and into the human clinical trial. And we believe by doing this, not only are we having a positive benefit on the reduction in the animal usage, we're also enabling higher quality candidates to enter the clinic, thus offering greater patient safety assurance. So in summary, again, we're very, very grateful to be highly commended in this particular prize. This is an overview of the paper. It was published in December 22 in Communications Medicine. And as I say, it is the first publication where we've gone through the IQ-MPS guidelines for assessing drug-induced liver injury. We've been able to replicate key histological structures and function within the liver. We can distinguish between pairs of toxic and non-toxic molecules. We also correctly identified clinical responses of additional drugs that IQ hadn't proposed that we included within our test set. And we believe that it does deliver a 3Rs benefit in the drug discovery process. And so with that, I'll close, but I just want to acknowledge some of my colleagues. Some of them are shown here in the picture. They're actually in the laboratory behind the glass, but it was very much a collaborative effort across Emulate. I'd also like to thank colleagues in AbbVie and J&J who performed the blinded assessment of the data for us, and also Dr. Jack Scannell who helped set up this concept of predictive validity and how one should really set up appropriate experiments to address it. And finally, to our scientific founder and a mentor of mine, Professor Don Ingber. And if you would like more information, please do use the QR code at the bottom of the slide. Thank you very much for your attention.
Thank you. That was a fascinating presentation there from Lorna. As Lorna mentioned, there was a QR code on her slide. So although she's unable to join us today, a recording of this webinar will be made available on the NC3R's website over the coming days. And if you do use that QR code, you are able to access a contact page to get in touch with Lorna and to ask any questions. I'll now hand back over to Julia to close the 3Rs Prize event.
Well, thank you very much, Natalie, and gosh, what fantastic presentations. Weren't we fortunate to have the opportunity to hear them? So huge congratulations to our winners again, and also a big thank you to all those people who submitted applications, because we really did have an amazing cohort of papers to review, and it's just so encouraging to see there is so much amazing work going on right across the globe in this very, very important area. So a big thank you again to my fellow panel members and Natalie for all the hard work that they've done to make this happen, and of course to GSK for continuing to support this important prize. As you may have realized, we are recording this webinar, and the recording will be on the NC3R's website in 2 or 3 days' time. So if you want to come back to it, hear the presentations again, you've got the opportunity to say so. But thank you to all of you for attending. This brings the webinar to an end, and I hope it's encouraged you to think about putting in an application, if not next year, in a year in the future, so that we can keep this very, very important work going. Thank you all very much, and bye-bye.